guys, welcome to today's video. Today we're going to be talking about ossification, and this is probably one of the more challenging concepts, so we'll go into a little bit more detail later, but let's just get some general concepts covered first. This type of ossification that we're talking about is also known as osteogenesis. Ossification just means the turning of something into bone, and osteogenesis, osteo meaning bone, genesis meaning beginning, means the beginning of bone or the formation of bone, the formation of the skeleton for the first time in a human embryo. So at week 8, the embryo skeleton is not actually composed of bone yet, it's just composed of fibrous membranes and hyaline cartilage, which are comparatively soft tissues. And around this time, this process of ossification also begins. So there are two types of ossification that we're going to be talking about: intramembranous and endochondral ossification. So the first type, intramembranous, is exactly what it sounds like. Intra means within, and membranous means of membranes. So intramembranous ossification occurs within membranes, and this is responsible for forming most of the flat bones in the skull. So that is the cranial bones, the frontal, parietal, temporal, occipital bones, and the clavicles. And all of these bones are flat bones, and this is the only way that membranes can be ossified, because the membranes themselves are flat and they retain that sort of same shape. And basically what happens in intramembranous ossification, again, we'll go into this in more detail, is that connective tissue, that's what CT means, connective tissue membranes are formed by mesenchymal cells, and then they undergo that process of ossification right there in that membrane. On the contrary, endochondral ossification relies on a cartilage model, and this forms most bones that are below the skull, so all of your long bones in your limbs, everything aside from your clavicles that's below the skull. And this happens by a replacement of the hyaline cartilage model by bone. So as we said before, in the embryo, most of the skeleton is formed by fibrous membranes and hyaline cartilage. And it is this gradual replacement, removing the chondrocytes and the invasion of the bone, that is what leads to endochondral ossification. So after you're born, obviously your bones still continue to grow. You grow taller, you grow older, and this is also a type of endochondral ossification. We will cover this in a later video, but basically your epiphyseal plates that consist of hyaline cartilage that's within your long bones, those actually grow and expand and get replaced by bone, and that's how your bones grow longer as well. So now let's take a closer look at each of these types of ossification. All right, now let's take a closer look at intramembranous ossification. So this was the first of the types of ossification that we were discussing earlier. So there are 4 major steps of intramembranous ossification. Here I've shown the first 2. Once again, these images are taken from Human Anatomy and Physiology, 8th edition by Marie and Cohen. So the first step here is that ossification centers develop in the fibrous connective tissue membrane. So this kind of bluish purplish tissue out here would be the fibrous connective tissue membrane. So what happens is that mesenchymal cells, mesenchyme is a type of tissue found in embryos. Stem cells from the mesenchyme condense into layers of vascularized primitive connective tissue. CT is my abbreviation for connective tissue. And then these stem cells will differentiate into osteogenic cells, also known as osteoprogenitor cells, and then osteoblasts, forming an ossification center. So as a whole, this kind of ball of cells is known as an ossification center, and these orange cells are osteoblasts here, which are derived from the stem cells. These osteoblasts, as we talked about in a previous video, secrete extracellular matrix. So the extracellular matrix is the osteoid and the collagen fibers that you see around here. And these osteoblasts eventually become osteocytes as the extracellular matrix hardens. So the second step of intramembranous ossification is the calcification of osteoid. Calcification just means the hardening of the calcium salts. So as you can see here, the osteoblasts are around the outside of the forming bony structure, and they're secreting osteoid toward the middle. Calcium phosphate and calcium hydroxide combine to form hydroxyapatite crystals. And the hydroxyapatite crystals form this needle-like structure, and that is the kind of palish color here that you can see, newly calcified bone matrix. And these combine with other mineral salts and ions to form a harder structure. And then eventually what happens is that these osteoblasts secreting osteoid around them, and then this ECM hardens, they actually become trapped within the osteoid itself. So that is when the osteoblasts stop secreting osteoid and they become osteocytes. So this is very similar to when we were talking about the microscopic anatomy of bone in the previous lecture. We were talking about how the osteocytes are the stellate shape. Here you can see that same stellate, kind of star-like shape, and they are residing in the lacunae with their extensions pointing outward. So this is just the beginnings of that. After that, here are the third and fourth steps. So in the third step here, Trabeculae and the Periosteum form. So the extracellular matrix is deposited in a kind of random fashion around these blood vessels, and that forms the Trabeculae, which make up the spongy bone in the middle of the bone. Spongy bone is also known as woven bone, so that's what you see here in the diagram, trabeculae of woven bone. And the trabeculae eventually forms the spongy bone around the blood vessels. The surrounding connective tissue associated with the blood vessels then differentiates into red bone marrow. And you actually see that in this picture over here. The diploid or spongy bone cavities contain red marrow. And then finally, the vascularized mesenchyme condenses on the outside of the bone. So all of this kind of bluish tissue will eventually condense to form the periosteum. Remember that peri means on the outside and osteum means on the bone. So the periosteum is the layer of connective tissue surrounding each bone. And then lastly, the compact bone replaces spongy bone deep to the periosteum. So on the outside of the layer here, you still see osteoblasts forming the bone and the trabeculae on the outside. So here, all of the bone is woven bone. It's formed by trabeculae, but then the outer portions here actually thicken and they condense and they become compact bone, which is also called lamellar bone because it's formed by lamellae. And the spongy bone persists in the internal portion, however. So you have the outsides being compact bone, the inner side or the inner part being spongy bone, and this forms the pattern called diploid, which is characteristic of the flat bones that we're talking about here. All right, now that we've explored intramembranous ossification a little bit further, let's talk about endochondral ossification. So because all long bones are formed by endochondral ossification, the example that we're going to see here is an example of a long bone. So the first step here is the development of the cartilage model. Remember that endochondral ossification depends on the use of a hyaline cartilage model. So we need to build this first before we can turn that into bone. So what happens is, this isn't necessarily shown in the diagram, but mesenchymal cells cluster and develop into chondroblasts. So the chondroblasts secrete cartilage ECM or extracellular matrix, which becomes the hyaline cartilage model. And the chondroblasts then differentiate into chondrocytes, and they reside in lacunae, but that's not terribly relevant right now. Then this bone collar, which is formed by periosteum, is laid down around the diaphysis or the shaft part of the cartilage model. So you see that here. Then in the second step, we see central chondrocytes hypertrophy and cartilage extracellular matrix calcifies. This isn't shown in the diagram per se, but the hyaline cartilage is composed of chondrocytes and they stop secreting collagen. Instead, they start secreting this substance called alkaline phosphatase. And this is very important as a transitional substance because it is needed for mineral deposition. So the collagen in the middle actually starts to break down and the surrounding extracellular matrix begins to calcify. So in the middle here, you can see the area of deteriorating cartilage matrix, and it's starting to be replaced by minerals. And this process is called calcification. The chondrocytes die and leave spaces called lacunae. And third, in this step, we see the development and expansion of the primary ossification centers. So the primary ossification center actually shows up as early as here. Basically what happens is a periosteal bud, which consists of nutrient vessels, lymphatics, nerve fibers, red bone marrow, osteoblasts, and osteoclasts invade the cavities that formed in the cartilage. So here you can see the nutrient artery, which is right now just the blood vessel of the periosteal bud. The osteoclasts will erode calcified cartilage matrix and make this hole even larger, whereas osteoblasts will secrete osteoid, the bone extracellular matrix, which will coat the cartilage to form trabeculae. Periosteal capillaries grow into the cartilage, which you see kind of happening here, and form a primary ossification center that spreads to both ends of the bone. So it starts in the center and then spreads outward toward both ends. All right, we're not quite done yet. We have 2 steps left of endochondral ossification. So in the 4th step here, we see the medullary cavity develop. And this is quite simple. As the primary ossification center grows here toward both ends, osteoclasts, which are bone-destroying cells, break down the spongy bone trabeculae here in the center of the diaphysis to form a cavity. And this becomes the medullary cavity as it fills up with red bone marrow later. In the 5th step, finally, we have secondary ossification centers. So while here we see the primary ossification center developing in the middle of the diaphysis, the secondary ossification centers will be found in the epiphyses, which remember are kind of the expanded ends of long bones. So what happens is the epiphyseal blood vessels, here is shown in artery, enter the epiphyses, and basically, the process is extremely similar to what happened in all of these steps over here with the primary ossification center. The only difference is that they exist in epiphyses and they kind of spread outward from the center of the epiphyses toward the middle of the bone and toward the outer edges. Finally, a thin plate of cartilage is trapped in the middle between the epiphysis and the diaphysis right here. This little stripe of blue is hyaline cartilage, and this is the epiphyseal plate. This is where your bone will continue to grow after birth, up until puberty or whenever you stop growing. And the secondary ossification center doesn't spread all the way to the very edge of the bone. A very thin layer of cartilage is left on the outside of the bone here and here at the 2 ends. And these blue crescent-like shaped things are the articular cartilage, which help soften the friction in joints. Thank you so much for watching today's video. If you have any questions or comments, please leave them in the comments down below, and we'll see you next time. Bye!